This is Dr. Lonsridge, and I am giving the last lecture for for general biology for this semester. And the topic is skeletons and muscles. And I have 10 slides. Miraculously, on the last video I said 15 minutes, and I was 15:09. So this one, I'm going to say 10 minutes. Let's see how good I am. All right, so this is the outline. Types of skeletons, mammal skeletons, and muscles. And actually, this last part about red and white muscles, I might not ask it any questions about it. We'll see how it goes. Okay, so begin. Skeletons, like what the heck are skeletons used for? First of all, what kind of skeletons are there? Well, there are hydrostatic skeletons, water-based skeletons like jellyfish and earthworms, and then there are outside skeletons, exoskeletons like in snails and insects. And then there are inside skeletons, which are called endoskeletons, that are skeletons of humans and other vertebrates. Hydrostatic skeletons, these kind, are good for small animals and then water. Exoskeletons are good for small bodies, and endoskeletons grow faster than body mass. And that's actually, I'm not sure if I talked with you about surface area to volume principle, but that's actually this is a good test question. Like, why does that have to happen? My guess is I have talked about surface area to volume principle, and so you can expect a question on this. And also, why are insects small? It's because the surface area, which is the skeleton, grows more slowly than the weight, which is the mass. And then what the heck do these skeletons do, right? Like, well, I'm going to focus on skeletons of the mammals because that's the most diverse set of functions. We begin with supporting the body, which of course it makes sense. It's also for holding muscles, for attaching muscles, and therefore for helping the body move. Mammals actually make red blood cells and white blood cells from the skeleton. Inside the skeleton, there's marrow, which is growing tissue that becomes different kinds of cells. And then there's also this part, which you do not need to know details about. The only thing you need to know is that this skeleton also is a place for storing calcium and phosphate. Calcium is illustrated here, but phosphate is also stored in the skeleton and the bone. Calcium is necessary for bone strength. Calcium is also necessary for body functions. So there's this give and take between what the body needs and what the skeleton needs, and it's one of the reasons why people need to take calcium supplements as they grow older, because they don't make as much, and if they don't make as much, then the body's going to start pulling it from the bone, and the bones get weaker as a result. Okay, we're moving on to the next thing. Now, the next thing is muscles, and muscles, of course, attach to skeletons. There are three kinds of muscles. I'm going to focus attention on one principally, but you do need to know all three, and this part right here is actually involuntary. I never change that. Involuntary and non-striated. I copied this over and I never fixed it. So non-striated or unstriated, I probably should say non-striated, meaning that there are no segments. So there's segments, you can see segments here, you can see segments there, but you can't see segments there. This is involuntary muscle. This is like the diaphragm. When you breathe, it contracts. Your eyes, when they dilate, this is your heart, and this is muscle that you actually voluntarily control, like your finger muscles when you want to move them. And that's what I want to focus the rest of the story on, is like how these muscles work. So the first thing we need to know is that muscles come in a variety of compartments or different levels. So we had a muscle, which is actually a bunch of cells tied together. Those cells are called muscle fibers. And inside those muscle fibers, so this would be a muscle fiber here. Inside those muscle fibers are these long tube-like things called myofibrils. And the myofibrils have inside of them, associated with them, the thing that's the contractile unit of the muscle cell. And that thing is called a sarcomere. So this is a sarcomere right here. This is a sarcomere right there. And they're kind of like a train just stacked up next to each other. Here's an example of a sarcomere. And that sarcomere actually has two components called the thick filaments and thin filaments that are made of two kinds of proteins, myosin protein and actin protein. Actin protein is just actually one of three proteins that are associated with this one. And it was like, what is all that about, right? Like, how does that work? Well, it works in a fashion that you will see in just a moment. But it starts with this. This thing right here is a sarcomere. And what you see, these dark bands, these blue bands, those are called thick myofilaments. And the thin myofilaments are the reddish bands. And so this whole thing from Z to Z is a sarcomere. And that is what contracts. And I'll probably post a video on how a sarcomere contracts just so you can see the dynamic nature of this. But here's the whole process. This is what you want to remember. So it starts with the stimulus. Okay, we got a muscle in our finger, let's say. And we got something we see, I don't know, you hear, maybe you hear me talking, you want to start writing. So you got an external stimulus. In this case, it's a car coming towards you. And then there's or it could be an internal stimulus that you want to text now, right? So the signal travels from the source, the eyes of the brain, through neurons. And you all know about neurons now, which can be several feet long, and they arrive at muscles, muscle fibers, muscle cells, right? And so this is a neuron. This is the axon terminal. There are synapses that connect the axon or the neuron to the muscle fibers. There's an electrical chemical signaling between the neuron and the muscle fiber, and that electrical chemical signaling involves neurotransmitters. And for muscles, it's principally a neurotransmitter called acetylcholine or ACH. Now, the third step is like what happens when that stimulus occurs. It triggers an electrical impulse to travel inside the cell to release a chemical called calcium. The calcium is stored in organelles normally when the muscle cell is resting. But when the cell has been stimulated, calcium is released, and that calcium then takes us to the next step. Calcium attaches to a thin myofilament and causes the connection to form between the thick myofilaments and the thin myofilaments. The head of the thick myofilament, myosin, which was in a high energy state prior to 4, releases that energy and causes the thin myofilament to move forward. This leads to shrinking of the sarcomere, and so it's like, kind of think of it as a ratcheting of a wrench. Repeat these steps 4 through 6 several hundred times conservatively. 16 strokes per millisecond. That's like 1,000th of a second. You get 16 of these strokes, these events. And all that time that it took me, about a minute to get through this, you would have probably had several thousand of these things happening. Here's a video that you might want to watch to show the animation of what is happening. And that, I promised you I'd try to keep it to 10 minutes, and I think I'm going to just do that. You could probably look at this. This is actually useful. I'm not going to tell you anything about it. There's white muscle, there's red muscle. Chickens have white muscle, geese have red. There are adaptive reasons why red is better in some cases and white is better in other cases. And that's a great test question. I'm not going to tell you. By the way, salmon have red muscle, but that's not really red in the sense of red muscle here. This is actually a pigment, a red pigment that these fish move to their skin when they migrate to spawn. That's a crazy thing. When you get the fish after it's spawned, that meat will be white because all those pigments have moved to the skin and turned that fish from a silver fish into a red fish. Okay, that's all. Thanks.